much for having me and for the invitation, and hopefully this will be informative for you, and I'm going to try to make it as painless as possible. So if you can see my slides here, we'll get started. The title of this talk is Medicinal Chemistry and Drug Discovery. I take this topic as my primary mode of employment, of course, very seriously, but as someone who sits through a series of very, very long and dull talks all day long at work, I try to inject some levity throughout. So if you see a meme or a cartoon and you want to laugh, go ahead. Let's not take ourselves too seriously here. This is supposed to be fun and informative at the same time. So without further ado, let me start in. I'm going to show here right on the title screen an example of some of the technological impacts that have occurred within my industry recently. This is an example of an X-ray crystal structure. You can see in light blue the small molecule, which is embedded in the green receptor, green and blue receptor. And this is the sort of technology which allows us to visualize how compounds are interacting with a specific biological target. This is sort of cutting edge technology. It's been increasingly used over the past few decades to the point where most projects are enabled in this fashion, and it's been very helpful to us. So let me go into a little bit of my background. Basically, as a high school student, just like you guys are now, I enjoyed high school chemistry, but in no way, shape, or form did I love it. I liked it. It was something I found interesting. Ultimately, what I wanted to do after graduating high school was to join the military, but unfortunately, I found myself, after securing a congressional nomination, medically disqualified for admission to the U.S. Naval Academy. They basically sunk that battleship dream real quick. After that disappointment, I started to re-engineer my ideas and dreams, and I was accepted at Georgia Tech as an undergraduate student. Now, when I selected my major, I had no clue what I wanted to do. And I think this is an important message to convey to all of you. When you're a high school student, you should not necessarily have to know what you want to do with the rest of your life, and that's okay. You can change your major in college. I changed mine 3 times before I even got there. So I started out as an aerospace engineering major. That industry basically tanked while I was applying, so I said, that's not a good future. I changed it over to nuclear engineering because it seemed interesting and very technical, but ultimately there wasn't much of a future there with regards to employment either. You'd either be working at a nuclear power plant or on a nuclear-powered submarine or aircraft carrier, and I didn't necessarily want to do that if I wasn't going to be in the military per se. And so I changed it to chemistry, and I said, again, I enjoyed chemistry in high school. This is something I might wish to pursue as an undergraduate. So I worked hard as an undergraduate, and I graduated with a BS in chemistry with highest honors. That's an important point. It was what enabled me to be accepted into UPenn as part of their PhD program. Obviously, getting into a graduate school, there's a lot of competition among students, and you have to basically achieve a certain standard in your undergraduate work to get into one of the best universities in the United States to study further. So after 3 years in the PhD program, I decided to actually leave with a Master of Science degree in organic chemistry. The asterisk just means I'll touch on that a little bit more later. That was actually a very, very wise decision, and I'll get into that on a subsequent slide. Went on to work for 5 small biotechs in the southeastern US. I'm a Southern boy. I'm got a lot of family down in the Georgia, North Carolina area, and so I wanted to work down there if possible. But all of those companies folded, and that's, again, what the asterisk is denoting here, that I'm going to speak more to this issue later. So I relocated the entire family to Merck West Point in 2009, where I've been a senior scientist since 2012, and I have about 22 years of experience in organic and medicinal chemistry industry, and I'm going to convey some of that to you guys today. So drug discovery. I'm gonna divide the talk into two major parts. First, a broad overview, high level of the drug discovery process in pharmaceutical companies, and then I'm going to talk more specifically about my job therein. But if we're covering just the general topic of drug discovery, it's typically divided into two phases. So you have your pharmaceutical discovery and your pharmaceutical development stages before you get a compound to market as an actual drug. I work in this orange sector, the discovery space. It starts out mostly with target identification and target validation being the responsibility of biologists and pharmacologists at any given company or academic institution. From there, chemistry starts to get involved as you start generating what we call lead molecules or lead compounds, and those compounds are then optimized to get the best possible compound, which is then brought into clinical trials where it's tested in humans. So the bolded text here is to emphasize the part where chemistry has the biggest role, and that's what I'll be focusing on. As far as my job, I'll go into the everyday tasks, the benefits, the challenges, and some of the cautions I have if you're considering this sort of role in the future. I should note that I have a big star above the target ID phase of pharmaceutical discovery simply because I was reading a paper recently of all the reasons that compounds fail in clinical trials to date. It comes down to two major factors. Number one is safety. Compounds that we put into human beings wind up being unsafe and causing a lot of adverse effects to the point where they're not approved to go to market. But the second and actually most important and most overwhelming reason compounds don't make it to market is because of target identification. So if a biologist identifies a target and hypothesizes that if we modulate this specific biological target, we will have an impact on a specific disease state, if that hypothesis is incorrect, all of the subsequent work that takes place winds up being lost time, lost investment, and ultimately very disappointing. And that's why it costs so much to generate a drug. I'll get more into that later. One thing I did want to start out with is that if you look in the mass media at pictures of chemists, you see baloney pictures like this one. This is the last thing a chemistry lab looks like. This lady isn't even wearing safety glasses. We don't work with these sorts of brightly colored solutions. Everything's either a black or brown or yellow sludge, basically. So don't allow these mass media representations of what I do make it look more attractive than it really is. So I'd like to start out with a broad overview of target ID and validation. Again, this is through the eyes of a medicinal chemist. I'm not a biologist, so a lot of this is, it's a skewed perspective. So in brief, as I mentioned, a biologist working with cells or isolates will study any given biological system and look for biological pathways which may have an effect on various disease states. This is, as I mentioned earlier, all very hypothetical. It's just a guess. It's an educated guess, and there's certainly demonstrable evidence to support these hypotheses, but until you get it into the clinic and test a drug in a human being, you don't know if you're actually going to impact any given disease state until it goes into a human being. So in other words, what happens if we want to divide this into two possibilities? What happens if you perturb a particular biological pathway by inhibiting any given target? Alternatively, you can try to excite a particular pathway by activating that target. Ultimately, what you want to know is that particular pathway druggable? In other words, can its activity be altered with a therapeutic agent? In my case, I work specifically with small molecules, but you can also envision biologics like proteins and nucleic acids doing the same sort of thing. Once this target's been identified, it must be validated. So the biologist needs to know, if I start altering the functional role of this target, do I see a change in disease state? Do I see a change in phenotype? In this case, phenotype, as we use the term, is a state of attenuated or lessened disease. This is best demonstrated, of course, if you use something called a knockout animal, where the target in mind is actually removed completely from the animal. So if you really want to see what the impact of any given biological target is, if you genetically engineer that target out of an animal, and then you try to inhibit this disease state and it doesn't work, then that's a very good indication that that target is going to have a big impact on that disease. Ultimately, you have to develop a cell assay, something that's easy, repeatable, can be scaled up exponentially so that you can test and screen what compounds antagonize or inhibit a pathway, and or alternatively, what would agonize or excite activity along any given particular biological pathway. And this is paramount for when I get involved, because if there's no way of testing which compounds are impacting the pathways, then we don't have a path forward. So in summary, I like to think of this as a faucet. You have a biological pathway, and let's say the faucet's open too much and it's causing a disease state. What you would want to do in that case is inhibit or antagonize that target such that you slow that flowing faucet down to a trickle. Alternatively, if there was a specific biological pathway which would be beneficial to a human being, you'd want to open that faucet up as much as possible. But again, like I said, I have a very skewed perspective on biology, something akin to this. I'm actually working on a bacterial project right now, and I just think this is sort of a funny picture, which chemists, we really don't understand biology, and if we pretend to, it's a sad state. So take everything I just said with a big grain of salt. So when does chemistry actually get involved? So when you finally have a cell assay available from a biologist, you start by screening a library of compounds to see which are impacting that particular target. And a library, obviously, the bigger it is, the better chance you have of finding what we call a hit molecule. So what you want to do is you basically design an assay, you automate the process, you screen upwards of tens of thousands, hundreds of thousands, or even in some cases, millions of existing compounds that you might have available to you in a library. And you see which of these random compounds is actually impacting that target. And assuming you find some of these hits, you can then verify their structures, make sure the purity of the molecule is sufficient, and then you screen similar molecules to identify what I'll call a hit class. And so what you see here is a bunch of phenylacetates with small structural changes, an isopropyl acetate versus a methyl, an amide versus a carboxylic acid, these sorts of things. And you start saying, I'm recognizing a pattern here, and now I know where I can start looking more closely. I chose these molecules randomly, but this first one, acetyl salicylic acid, that's aspirin, so that's an actual drug as an example for you guys. This process, obviously, with these big libraries of upwards of millions of compounds being screened, is highly roboticized. And so I just ask you guys the question, as we head into 2021 and beyond, is a robotics engineering degree more valuable than a science degree? I would argue that perhaps it is. The robotics industry is taking over multiple industries, and it's certainly having a huge impact on the pharmaceutical industry, so keep that in mind. So chemistry then takes over. Once we've screened these millions of compounds, we have our weak inhibitors, our hits. We identify structural motifs that are similar, and we start breaking down the molecule into its various parts, looking for a strong inhibitor, which we would call a lead. I put magic in the arrow, but it's not really magic. This is actually just the process that a medicinal chemist goes through, whereby he or she can identify what aspects of the molecule lead to an increase of activity against the target in mind. And so you can see that altering their hit molecular structure bit by bit, identifying what role each position plays, all while monitoring for increased activity in the cell assay and maintaining drug-like properties. So you can quickly blow these sorts of small, simple molecules up into increasing complexity. But as you add molecular weight to any given small molecule, you're becoming less and less drug-like. You're decreasing your aqueous solubility, so it's not going to circulate in the blood as freely. You're possibly increasing off-target effects, which lead to all those adverse events which you hear described in any given pharmaceutical product commercial. you could start seeing toxicity creep in, et cetera, et cetera. So this is an iterative process, and I'll describe that more in a second. The difficulty with this is that organic chemistry allows for a multitude of molecular transformations. There are literally hundreds of often used name reactions that we use over and over and over and over and over, which allow us to transform simple starting materials into more complex products. And we can then screen those in a biological assay. But sometimes if the compound that we want to make is sufficiently complex, the toolbox winds up being empty, and then you have a chemistry problem. Then you have to figure out a way to make the compound that you want to test in the assay, and that can be very difficult. In the case of these compounds down here, it's very simple to make these from commercially available starting materials, of which there are tens of thousands available for order. I refer to these as Mickey Mouse chemistry. Take a look at that hideous thing. That's nightmare fuel right there. Alternatively, if you want to look at the other end of the spectrum, such that the synthesis is extremely difficult, you can look at a compound like palalamine. This took 25 steps to make and 0.015% overall yield. And just to put that in perspective, if you were to start with 10,000 molecules of a simple starting material, by the time you make palalamine, you've got 2 out of those 10,000 molecules actually converted to the product. So these are the 2 extremes, Mickey Mouse and extremely difficult. The chemistry I do is probably residing somewhere in between, but obviously the closer you can keep it to simple, the more products that you can screen and the better data that you'll generate in a more rapid fashion. So we try to keep things as simple as possible from a synthetic chemistry perspective, but also it helps with some of these other off-target effects. So lead optimization. Once we've got a really good molecule, we need to know, is it the best molecule we can ever make in this particular space? I ask the question, is more data always better? This is a screenshot, one of many screens of data for a single compound that we generate at Merck. You can see here I've had to block out all the assay names, descriptions, and the structure because I'm not allowed to divulge that to you guys. But what I wanted to illustrate here is the dozens and dozens of data boxes that are filled for any given compound. And what we're trying to do is balance all of these properties to narrow down to one single compound, which has the best overall profile. So if things like solubility, we'd want aqueous solubility to be high. Potency, we'd want to have the most potent compound possible, et cetera, et cetera. Balancing these properties is not simple. And as I mentioned, this is one of several pages of data that we'll generate on any given compound. We actually have entire computer programs devoted to helping us visualize and parse this data because it gets so complex trying to track everything that's going on with these molecules. So that brings me to the weekly process of a medicinal chemist, and that's looking at making compounds, submitting them to the cell assay, and then using the data combined with various tools to sort of assess if you're making progress on your particular biological target. So you see here, we generate these compounds every week at Merck. We call them L numbers. Each compound gets its own unique L number. We submit it to our biological assay, which again, I can't tell you what they are for this particular project, but we generate something that's called an EC50. This is the effective concentration at which 50% of the target is, in this case, inhibited. So obviously what you're looking for here is the concentration at which 50% of your target is inhibited to be as low as possible. So in this example, we have one compound with a 781 nanomolar concentration at which 50% of the target is inhibited, and another at a higher concentration of 1560 nanomolar. Basically means compound 1 is about twice as potent as compound 2. And week after week, month after month, year after year, this is the sort of stuff that we're looking at, comparing compound A to B to see if we're making progress. What are the structural motifs in this compound which are unique such that they're engendering this greater potency relative to structure B? And it's sort of like playing molecular detective. You come up with a theory, you make the compound, you're testing the theory in the assay, and you move on from there. And that brings me to the final stage, which I said I'd touch on briefly, but I'll spend a little bit of time here. Clinical entry. So after upwards of, in some cases, decades, we're about halfway to market once we get to clinical entry. So in our example, let's say this was the actual final molecule that we selected as the best that we could possibly come up with to hit our particular target. This is the greatest potency, has the least off-target effects, and it has the best what we call PK or pharmacokinetic properties. Pharmacokinetics is a study of what does a body do to a compound. So if you were to give this to a human being, what does a human body do once it's inside? How does it break it down? How fast is it cleared? In other words, how fast is it no longer present? And things of this nature. That's a whole subsection of chemistry and biology. It's extremely complex. So basically what we do is we nominate this compound for advanced development. Another team will then start looking at the molecule with greater scrutiny, but also in different ways. So they'll look at the best way to synthesize it in bulk. They'll look at the best way to formulate it such that they can develop a route of entry. So it might be an inhaled drug, it might be an injected drug, it might be something that you swallow in a tablet or a capsule, it might be a transdermal patch. These teams will look at these different routes of entry and try to develop the best way to get it into the human body. Ultimately, another team will look at how clinical studies should be designed. So just touching on clinical studies briefly, there are basically 4 phases, but technically you could break it into 5. So your phase 1 study consists of healthy volunteers. They have no disease, they're not suffering in any way. They're just there for evaluation of the compound from a safety perspective. Also use the data that they get from these people to establish what we call a human half-life. So again, touching on the PK or pharmacokinetic properties, how long is this molecule actually in the human body? And we calculate what we call a half-life. How long does it take for half of that drug that's given to be eliminated from the body? And that gives you an indication as to what sort of doses you should go into phase 2 with, how often the human being should be dosed, et cetera, et cetera. Once you have that data and everything looks safe, you go into a much larger phase 2 proof of concept study. So here we're looking at people with the actual disease, and we're looking to see, is the drug altering the biological target? Not necessarily the disease state, but just the target. So an example I would use is in cardiovascular disease, are you affecting a biomarker like LDL? Are you lowering LDL with a compound like Lipitor? That's basically all you want to know in phase 2, but you're still looking at half-lives, you're still looking at safety. In some cases, unfortunately, we have to run what's called a phase 2B study, and that comes up when this phase 2 study, phase 2A, winds up being insufficient to prove that you're altering the biological target to a reasonable extent. So you have to go in with a higher dose, maybe a longer duration of study, maybe a larger study to get to statistical significance of the effect you're looking at. Finally, you'll go into phase 3, a very large scale trial with lots of patients, usually very long studies, and you add final proof the compound is safe and efficacious, and you do this via a specific predetermined endpoint. So in the case of Lipitor, whereas in phase 2, you're looking at lowering LDL, in phase 3, you're actually looking at the disease state itself, and you want to say, yes, my compound is efficacious to the point that it's lowering the frequency of heart attacks, something like that. So obviously, not a lot of people have, in a random selection of cross-section of people, not a lot of people will drop dead from a heart attack, so you have to have a massive amount of patients such that you reach a sufficient number, unfortunately, who will have a heart attack when they're on a placebo versus the group that are on your drug will not have a heart attack. So you have to have enough of a difference in numbers of those two groups that you can say, yes, the drug is in fact working the way you want. There's actually a fourth phase to clinical study that takes place after a drug's been approved. It's usually to look at more safety data, and it also can be used to look at different indications. So an example I would give there is something like Humira, which is the best-selling drug in the world, going on for several years now. It was originally approved for rheumatoid arthritis, but after being approved for that indication, they took it into phase 4 studies, and they looked at things like psoriatic arthritis, which is a smaller market, and they now have approval for both of those forms of arthritis. Ultimately, take-home message from this page is that even before we get to clinical entry, this is a very long, very expensive process. And so, I love y'all's meemaws and peepaws, but when they start complaining about how much their drugs cost every month, it kind of rubs the medicinal chemist or anybody in the pharmaceutical industry the wrong way. This can take decades to get to market, and the estimated cost as of March 2019 is $2.6 billion spent to get a single drug to market. If that drug is only selling upwards of $100 million in revenue each year, you can imagine how long it takes to try to recoup that investment. If a drug recoups that investment more quickly, people like me, we're not getting rich off of it. We're simply in a situation where the company will then reinvest those dollars looking for the next drug. And so that's why pharmaceuticals are sometimes expensive. It takes a lot of money to recoup for all this investment and time. So the job, this second part of the talk, I'm just going to talk about what I do specifically. What are the day-to-day duties of someone like myself? Now, the first thing is safety. You're not supposed to set yourself on fire, or others on fire, and goof around in the lab. This is an old-time favorite of mine from Far Side, Gary Larson. We work in a very unsafe environment. Obviously, engineering controls are in place to try to prevent people from getting hurt, and we use personal protective equipment out the wazoo to try to keep people safe. But it's very important because we work with a lot of toxic and highly flammable reagents. So some of the hazards I work with on a daily basis are things like inhalation hazards. I work with a lot of gaseous compounds, which are, if inhaled, can make you very sick or ultimately even kill you. Pyrophoric reagents, these are reagents which, once they're exposed to air, will suddenly burst into flame. That's very exciting. We work at a lot of very high temperatures, sometimes in excess of 300 degrees Celsius. So we're talking skin melting temperatures. We work with flammable solvents, and we work with them in bulk. It's to the point where if you were to drop, say, a 1-liter bottle of something like diethyl ether, what will happen is the vapors from that ether will float around in your space until they hit an ignition source. It could be something like an electrical switch. And they will flash to the point where that entire 1 liter of solvent will either explode or catch fire very rapidly, and fire starts spreading like you wouldn't believe in a chemistry. I've seen entire chemistry labs destroyed. And in fact, about a month and a half ago, Merck lost upwards of about $15 million of lab equipment in the lab just below mine because a fire broke out and destroyed basically the entire space. Luckily, no one was hurt. It occurred after hours, but that's something we always have to keep in mind. We work with a lot of strong acids and bases, which will burn the skin, much like poor Professor Bingman's neck back here. And special mention of robots. Again, occasionally you run into a nice friendly one, but for the most part, they're like the Terminator. So unlike, say, the robot we all have in a lot of our homes, a garage door opener, which has a force feedback loop such that if the door starts to close and there's a child or an animal in the way and it hits that subject, for lack of a better word, it'll identify that stress in the mechanism. It will stop trying to crush them to death, and it will actually reverse. None of the robots I work with do that. All the robots I work with try to kill you. They basically have no force feedback loop. And so if any of you do go into the robotics industry, please try to keep that in mind as you design, because I've almost had my arm taken off a couple times, and it's quite unpleasant. Going on to other parts of the job. So I've also already mentioned the week-to-week process, but to go into a little more detail, we look over what we call existing structure-activity relationships every week. So we look at those data sheets that come with the two compounds I showed earlier. We're comparing which compound is more potent relative to the other, which is hitting the biological target more effectively. And we propose a novel idea to alter that structure. How are we going to change it such that the theoretical new compound will improve that existing chemical matter? We don't know if it will. There's no way of knowing ahead of time in most cases until you actually make the compound and have it screened in the assay. The next step is to figure out how do we make it, and we use something called retrosynthetic analysis. I have an example of that here. So what you do is you say to yourself, I want to make this compound. How am I going to make it? And you start saying, well, if I have this, I know I can make it from this precursor. I just do a diethylation on this aniline nitrogen. If I have this substructure, how do I make the preceding intermediate? Well, I know I can reduce a nitro group to this aniline nitrogen, and so on and so forth until you break it down to very simplistic starting materials, which you can order from any number of chemical vendors. Sometimes this works very well and you get all the way to the end in one shot. And sometimes you have to sort of reorient your synthetic route and maybe do a completely different synthetic route. AFO cyclization, I put the acronyms definition here. This is one of those name reactions. And just to give you guys an indication as to my level of expertise, I have no idea if such a reaction even exists. I don't typically work with what's called a chalcone or a flavone, but evidently this is well-known chemistry in this space, and unless you were working here, you probably wouldn't know about it. So in order to do this efficiently, they actually have a lot of computerized tools available to us nowadays that they didn't have decades ago, wherein you can input, say, a part of the structure, maybe just this phenylamine. And you can say, how do people append ethyl groups onto this? And that computer program will tell you who's done this sort of chemistry, what their substructure looked like, how they did it, and then you can look up the paper and you can get a procedure to do it yourself. So we have a lot of help thanks to information technology improvements over the last couple of years in order to generate these sort of retrosynthetic analyses. So once we know the route we want to try to get to work, we order the starting materials. We actually begin what we call wet chemistry. That's the real fun. It's where we set up, monitor, work up, purify, and characterize our products. And then we wash, rinse, and repeat, oftentimes for years, this process. So we'll start, again, looking at the assay data, proposing new targets, figure out how to make them, get the starting materials, make them, et cetera, et cetera. So just to show you where I work, this is my fume hood at work. I mentioned flammable vapors earlier. Fume hoods act under negative pressure, so they're constantly pulling air out of this space such that if you do have flammable vapors in this space, they aren't hanging around. They're actually being pumped out through the ceiling and out through the roof of the building so that they don't catch fire or explode. Opposite this fume hood is my bench. It's a little cluttered at this time. You can see all the different types of tools and equipment and supplies that we need to do the basics of the job. And this is me with my ever-increasing receding hairline and graying hair. This is a job that will tax you. So as I mentioned, every day we come in, we run reactions. We don't typically run just one. We'll identify perhaps a whole bevy of targets that we want to go after. We mix them up, we heat them up, we pressurize them, whatever is necessary in order to do the actual chemical reaction. These all occur in solvents. Obviously, you want to dissolve the compounds that you're trying to get to react together so that they mix well and they interact. It's two solids mixed together don't typically react with one another. As such, once you're done with your reactions, you've got to work them up. You've got to strip your solvent away from what is hopefully your product. This instrument is one I use every day. It's called a rotovap. It's simply a heated water bath connected to a vacuum pump. And by heating the solution and applying a vacuum, you actually pull the vapors of the volatile solvents away from the product Those are condensed in ice traps, and you move on to the next step, which is purification. Here's one of the robots that tries to take my hand off all the time. This actually is a purification instrument where we put a silica gel column here, and we actually pump our compound mixture through the column with different solvents. And as different things come through that column, this robot automatically collects them as different fractions such that you can separate one compound from the next. So, job part 3, why chemistry is better than biology. I love this part. Ultimately, what makes chemistry better than biology to me is I always know what I've isolated. Biology is not always the case. So one of the tools we have at our disposal is called LCMS, or liquid chromatography mass spectrometry. When we're running a reaction, we can actually take a small aliquot of that solution. It's basically like a little pinprick amount of solution. And we can inject it on an instrument, another roboticized instrument, an LCMS, and that will actually push our compound with various solvents through a column. And depending on the characteristics of what's packed in that column, the characteristics of the compounds that you have in your reaction, they'll actually come out at different times. So some compounds will come through the column quickly, like this one. This little peak indicates a single compound. That's because it's not retained on the column. It prefers to stick around in the solution you're pumping through the column. Whereas other compounds that you've got in your mixture will retain themselves on the column longer, so it'll take longer to come out. Now, this alone gives you some indication that perhaps if this was starting material and this peak gets smaller, this might be product. But if you couple this instrument with mass spectrometry, you can say, what masses of compounds are in this green peak? And let's say 486 was your target, you have a very good indication that you've made the compound you want to make in this solution, and now it's simply a matter of purifying and isolating that single peak to bring it to the next step. That next step, once you've isolated that clean compound, we put it through something called an NMR, nuclear magnetic resonance spectroscopy instrument. This is basically the equivalent of an MRI machine. Uses different levels of magnetic fields to characterize your compound. So how do you know if you have the right thing? Well, you've got a molecular weight from the LCMS, and now you put your sample into this instrument, and depending on where the protons are in your compound, you get shifted peaks, and you get different splittings on the peaks. And I could teach a 3-day seminar on how to interpret this, but overall, all you have to know is it tells you how many protons are in each peak. So you can add those up, make sure they're commensurate with the target you're trying to make. And you can look at the relative shifts and say, okay, this looks like the methyl peak I wanted. It's got 3 protons, same as this one. And you can identify beyond a shadow of a doubt in 99.9% of cases that you have the correct compound. So again, why is chemistry better than biology? At the end of the day, I know what I put in the vial. I have no doubt that what I put in there is the right stuff, and I like that sort of certainty. I don't think I could be a biologist because I don't like uncertainty. So keep that in mind if you're interested in biology. I'd like to finish up here by just describing the industry as a whole. I'd like to start out with some of the benefits. So every single day, both your mind and body are engaged and challenged. This is a very hands-on type of industry. Doesn't matter if you're working as a biologist, if you're working in pharmacokinetics, if you're working as a medicinal chemist, you're going to be running experiments, and that allows you to engage both mind and body constantly. Other than medical or biological research, in my opinion, medicinal chemistry is probably the most technically challenging profession on earth. A lot of people use rocket science as an example of something technically challenging. But when you're building a rocket, you put the thing together and you test it, and you know within minutes whether or not it works. In medicinal chemistry, it can be decades before you know if what you're working on works. And again, it has to do with that long timeline to get something into the clinic and through the clinic. So it's impossible to describe the challenges that we face in this profession, and trying to impact human health, which is something else positive about that, while overcoming what we'll call very complex problems. So a benefit of that is you get to work with teams of half a dozen up to many dozens of people who are very intelligent in their particular field. And it builds a real uh, team spirit as you go through, work through these problems. You get to constantly present your work and results. This can be either in written or oral fashion. You're constantly giving updates by email, formal presentations, or even in group meetings. You're trying to capture key data and whatnot in publications and submitting them to journals for review and publication. So that's a huge benefit. There's constant on-the-job learning, so that the industry is never stagnant. Once you get in there, it's just the beginning. You're going to learn a lot on the job, much more so than most industries. And it has good pay and benefits that go part and parcel with it. Most important message I can leave you with today is that this industry absolutely needs hardworking, motivated, and intelligent people. And I hope some of you will consider it in the future, because human health depends on you. The future of human health is the kids who are in high school right now who might be considering science. There are challenges, however. I'll go over these very briefly. Engaging the mind and body every day can become exhausting. I've been in this industry for over 2 decades now, and I'm getting more and more tired every year. Not getting any younger. It becomes very tiring to the point where, you know, things start to ache and groan in your body that didn't when you were younger. It's kind of like an athlete that is past his prime. Very few projects have a positive outcome. This can become very frustrating and actually drives a lot of people out of the industry. They don't have the patience to overcome problems. They don't have the patience or the wherewithal perhaps to deal with the constant disappointments. And there are constant disappointments. I've been working, like I said, 2 decades in this industry. The furthest I've ever got a compound is into a phase 1 clinical trial. So you could basically say everything I've worked on has been a failure to date. Hoping for some success in the future, however. Talked about working with teams. Unfortunately, especially at some of the larger company, lots of scientists, present company excluded, of course, are absolute weirdos, and they're not good at personal interaction. So you have to be on your toes and learn to work with people who might not be the easiest to work with, let's say. Presenting your work and results is fine and well if your public speaking skills are up to snuff. I tend to trip over words and I'm a mush mouth, so I have trouble here, but you develop this skill the longer you're in the industry. You're constantly asked to present to groups of 10, 50, or hundreds of people, and so you have to be willing and able to develop that skill. On-the-job learning is great unless you start to struggle keeping up. I haven't read any of the scientific literature in probably the last 5 years because I'm so busy with my particular work. So if it doesn't pertain to what I'm working on, I don't have time for it. And that means I've fallen a little behind, and you just have to be able to deal with that as well. Good pay and benefits only go so far. It depends on the company, the size of that company, and recognition of your efforts, of course. So all these things are challenges. I will give this last slide a few very serious cautions to you. First caution is, what level of degree do you need to work in the pharmaceutical industry, or more specifically, medicinal chemistry? Bachelor of Science, Master of Science, or Doctor of Philosophy? What is required? It's not really a question I can answer with any large amount of certainty. I will tell you there are no Bachelor of Science chemists in my entire department. We used to hire Bachelor of Science chemistry graduates straight from undergraduate education, and then they just stopped hiring them altogether. We're also not hiring any more Master of Science chemistry graduates who have some graduate school experience. All of our hires lately have been PhDs. Now, if you decide to go to grad school and pursue either a Master of Science or a PhD degree, just keep in mind that's a process that's unique. It takes typically 4.5 to 6 years to earn a PhD at any given institution. During that time, you'll be doing research, you'll be teaching undergraduates, et cetera, et cetera. What's important to keep in mind is there's an ever decreasing market for all 3 of these levels of graduate in my industry. If you get a PhD, what's most important is where did you earn that PhD and who did you work for? My company, my department has not hired anybody outside of, I think, 4 different universities over the last 10 years. So if you don't work in those, if you don't do your graduate work in one of those 4 institutions, you're probably not going to get a job at Merck West Point. So you have to keep that in mind. Moreover, if you get a PhD at a different university and it's not a very good one, let's say your research isn't very compelling, you're looking at a postdoc study. Those are usually 1 to 3 years, and it's basically like an extended PhD program. You go, you work, you do research for someone, you earn a small stipend, but it's typically like on the order of $20,000 a year, and you could be doing that indefinitely until you find a job in industry or decide to go into academia and teach. One of the biggest cautions I can tell you about is consolidation leading to fewer jobs in the industry overall. So that when I first got out of school, out of Penn in the year 2000, I could typically procure anywhere between 5 and 10 interviews for job openings. Nowadays, if I wanted to look for a different job, I'd probably have between 1 and 3 opportunities to interview. There's just not as many small and medium-sized pharmaceutical companies around anymore because the big boys have swallowed them all up, because it's really hard to design and discover new drugs. And so you see a small company that has a good idea, you simply buy them out and you get all their intellectual property and you develop the drug for them. That's something that's unfortunate, but I think it's hitting not just this industry, but others. Another huge caution, reorganizations in my industry have led to huge layoffs. As an example, at the end of 2013, 67% of the people in my department were laid off. They were not rehired. They basically had to go find jobs elsewhere. This sort of reorganization and layoff paradigm has led most of the people I've worked with outside of the pharmaceutical industry. They either go into sales or they go into something altogether different. Last thing is, this job is extremely outsourceable. Part of the reason there's been these mass layoffs in the industry is because all those jobs, the 67% of my coworkers who were basically let go, all of that work was then sent to what we call external resources. These are chemists who work in China and India at a much lower pay rate than what we earn here in the United States, so they're much cheaper to hire. Chemistry is extremely outsourceable. I could take anybody in high school and I could train them how to do my job. They won't know all the details, they won't know all the ins and outs, but they could do the basics of what I do. And that would probably take me a year to train someone to do that. So because of the outsourceability of this position, I caution you against blindly leaping in and saying, this is absolutely what I'd want to do with my life. It becomes, there's a lot of hurdles here that you have to constantly jump over to maintain yourself in the industry. Last thing, and I'll close this. This is something I wish somebody had pointed out to me when I was pursuing an organic chemistry degree in graduate school, or even as I was pursuing a chemistry degree as an undergraduate. Where are the jobs for this particular industry? I had no idea. I said most of my family is located down here in the Southeast. I had no idea most of the chemistry jobs are located in 2 spots of the United States. Massachusetts, specifically the Boston-Cambridge area, and California, specifically the San Fran Bay area, and perhaps down into San Diego as well. There's 2 things to point out here. These are both very nice places to live. And the second thing is, these are the most expensive places to live in the entire continental United States. Keeping in mind where you're going to work, where you might find a job, it's the kind of thing that the typical guidance counselor won't have that information available to give you, but it's definitely something you should look into before you start pursuing any given mode of study or potential for working in industry. And so with that, I'm happy to take any questions you guys have, and I really appreciate your time and attention today.
I would like to just say thank you so much. That was unbelievable and the most informative probably presentation I've seen in a while. So thank you
Oh,
so
thanks.
much for that. Um, I guess the question I had, it really just went back to earlier in the presentation. It seems like a lot of that process, that original like target identification process, it's a lot of thrown at the wall, stuff at the wall and seeing what sticks. Is that pretty standard through like the pharmaceutical chemistry field, that it's in the sense that, yeah,
Yeah.
yeah, like that early target, when you were going through the process of doing all the assays and trying to identify, with the library, is every drug kind of approached that way, where it's this huge scope initially and then it kind of gets pared down?
Yeah, I'd say probably 90+% of our projects operate under that paradigm where it's sort of like a giant funnel, and you start as broad as possible and you try to narrow things down, because we're not at the point yet. I showed this crystal structure in the title screen. We're not at the point with computers yet where we can ask a computer what sort of molecule would fit into this particular target the best and give us the best potency. That's just an impossible problem for computers at this stage to answer. So as you mentioned, throwing stuff at the wall and seeing what sticks, that's 90% of our industry doing that. It's coming up with that hypothesis, trying to develop the assay, seeing if there's any compounds that hit that particular target, and then trying to get the best molecule possible. And as an example, I'd say over the last 11 years I've been at Merck, I've worked on probably 10 projects, and many of those didn't make it much past this stage, right? Early lead optimization, just because we found problems. There might have been an unidentified toxicity problem. There might have been a lack of efficacy against that particular target with these molecules, and the whole thing just winds up getting scrapped, and you start over with a new project. So the biologists at the very first stage gate are constantly trying to find and identify new targets, but they have no idea if it's something viable, if it's something druggable. Ultimately, will it work in human beings, and will it make it to market? So this big star being the most important part of the process, it's 99.9% guesswork, unfortunately, at this stage. And it leads to that long timeline and the huge expenses to get something to market, because when Mima and Peepaw, I made the joke, are paying for their drug, they're not just paying for the drug that's actually being bought, they're paying for all the failed attempts to generate a drug as well. You've got to recoup those research costs somehow, and unfortunately, that's the only way to do it. But that's a good question.
And I also just wanted to thank you on a personal level for your job, because I just started on a targeted kinase inhibitor, and yeah, thank you.
Yeah, thanks.
I had a question about specifically, you earlier mentioned briefly about robotics and how the overreaching industry might have like a future impact on your job. So do you have any like opinions or predictions about how that could affect your job and just generally like the pharmaceutical and healthcare industry in general?
So for me specifically, there's actually been a lot of development into synthetic robots, to the point where companies are trying to develop automated systems that do 95% of what I do, which of course is very scary as a medicinal chemist, because I don't want Bender or the Terminator taking my job away. I'd like to be able to support my family, and it took a lot of education to get to this point. But that's sort of pie in the sky type stuff right now. It's not viable. And you might imagine a robot would have a hard time handling a mixture of gases, solids, liquids, gums, and snots, and all this sort of stuff that we have to scrape out of round bottom flasks and vials and whatnot. So I continue to hope that that will be a struggle and that won't impact my job specifically. As far as robotics in the overall industry, it's having an impact on every single facet of drug discovery. There is nothing that we do that has not been roboticized or is not attempted to be roboticized, to the point where much of what we do is not hands-on work, it's simply managing the robots. The better the robot, obviously, the easier it is for us to manage it. And so a lot of that comes down to software implementation as well. So that's also a huge field in the industry. And the two or three, in this case, software, robotics, and pharmaceuticals, that these three industries working together is absolutely vital for us to shorten the length of time and the cost it takes to get a compound to market. Because only by working together will we develop the fastest routes possible. And I think you're seeing some of the impact of the investments over the last 30 years by the rapidity of the vaccines for COVID coming along. These things were developed at breakneck speed, and it's only because we have access to some of these newer tools and technologies that we didn't have before, and it's making it faster and easier to get to market.
Okay, thank you.
Yeah.
I have a question too. So you mentioned before that when you were in high school, you like chemistry. What advice do you give to high schoolers who are struggling to figure out what they want to do in college? And do you think that we should kind of analyze the classes we take now and see what we like, or should that realization come naturally?
Oh, that's a tough question. Ultimately, if I had it to do again, I might not have declared a major as I entered my undergraduate studies. The unique thing about college in the United States is when you get to school, you're going to be required to take any number of classes which are completely unassociated with your declared major. Things like English and math, which may or may not be associated with what you do, history, things like this. These are all requisites to get an undergraduate degree. So theoretically, you could go to college not knowing what your major is, not declaring on anything specific, start taking those requisite classes, get them out of the way, all the while exploring what is it I want to do, what does the job market look like, where would I ultimately have to live, things of this nature, which you might get more feedback and insight into in a college setting than you would in a high school setting. Maybe. It's not a guarantee either. But certainly asking other students at college what they're studying, what they're enjoying, what is it like, how do they envision their career, what do they envision their job being like, these are probably the next best step to simply interviewing and asking questions of someone who's already in the industry. So that's why I was trying to convey as much as possible about what I do on a daily basis to show if you don't like standing in front of a fume hood and a chemical bench all day long or most of the day, this isn't the job for you. But that's the day-to-day tasks that I do. And the only way to know that is to ask someone who has that particular job. So I think job shadowing is crucial. I think asking people who are in the industry is crucial. And I think maybe deferring on a major is useful to a lot of high school students. A lot of times we like to choose what we're going into school for before we even get there, like I did. And I don't think that's necessarily the best way.
Okay, thank you.
You're welcome.
Do you expect that for job prospects to get any better, or do you think that this trend will continue?
So in the pharmaceutical industry, it's interesting because whereas my job is highly outsourceable, other aspects of the pharmaceutical industry are not. So most of our people working in something like pharmacokinetics, which I mentioned, it's not as easily outsourced because it's a real specialty. It can be difficult to get those jobs because there's not as many of them per se out there in the United States. But once you're there, you're probably not going to get laid off as readily as I am. I've been laid off twice in my career, and it's very unpleasant. You're suddenly cast into a situation where you have no income other than the severance package you're given, and you're in a panic to find a new job. So you want to try to avoid that kind of scenario as much as possible. As far as biology, I also don't think that's as outsourceable because those are the people who are coming up with the novel ideas as far as what to target and what projects should be initiated. And that's not the sort of thing that you'd outsource from the industry. Chemistry is unique in that like I said, anybody could be trained to do the job, and as long as you're safe and you know what you're doing, you can make compounds. You might not make them as efficiently as somebody with my level of experience and hopefully knowledge, but you can make them, and certainly that's what we've seen. We've seen jobs from my department outsourced, and the chemists in China and India that we use, they are increasingly efficient at what they do. And fortunately for American scientists, they're becoming more and more expensive. So I look forward to the day when wages in India and China are on par with those in America, such that American companies don't want to outsource my job anymore. So that's the best insight I can give. I think the industry overall is never going to shrink beyond a certain point, and maybe we're at that point. Maybe there is a bright future ahead. There are certainly a lot of unmet medical needs. I think the fight against things like Alzheimer's and Parkinson's disease and some other neurological disorders is pain as an indication, chronic pain. These are hugely unmet medical needs, and the industry's desperate to try to solve these problems. And so far, we've been completely unsuccessful in most cases. So there is still an impetus to improve human health, and there will always be opportunities. It's just a question of getting the proper education at the proper place, being competitive with others with the same level of education, such that you can get an interview and get a job, and then making yourself as important as possible in that job by doing constant good work and trying to keep up with the state of affairs in the industry and in the particular disease state that you're studying.
Okay, thank you.
Well,
Any more questions for Mr. Skudlarek at all? Okay.
Okay, if there are no more questions, then I think we can end the meeting here, but I think we should all first give a big thanks to Mr. Skudlarek for coming in and talking to us. I know that this was definitely an extremely informative guest speaker session, so thank you for that.
thank you. And if you guys have any other questions, Regan has my email. Feel free to reach out. Hopefully I have enough insight in big pharma, mid-sized pharma, and small pharma to answer most of the questions you might have. And if I don't, I can reach out to some of my colleagues who might be able to answer specifics about their departments as well. So please reach out if you have other questions.
And I also just wanted to add that in the pharmaceutical companies, there are many different job opportunities. So there's chemists, obviously, biologists, and then, like, my wife works in the regulatory affairs department, which is, she has a pharmaceutical chemistry degree, but she went an entirely different route. So pharmaceutical industry in general, though, is a great industry to get into financially and just job security. Well, other than, I know you just talked about the layoffs, but
Yeah, that's
But-
mostly the medicinal.
Yeah.
Regulatory typically isn't hurt by layoffs because that job is absolutely mandatory
Mhm.
in order to keep compounds moving through clinic into approval of market. So that's an interesting thing to mention. That the pharmaceutical industry is so broad and so vast and touches so many different technical skill levels that there's something there for anyone and everyone who would want to be a part of it. And like I said, take nothing else away from this presentation, please take away, we need people who want to work in this field. Absolutely, because we don't want to fall behind. We want to get in front of some of these diseases and try to impact human health as soon as possible.
Are you allowed to say what diseases you've worked on?
So I've worked on everything from the cardiovascular space, heart disease, to the neuroscience space, things like schizophrenia, to the infectious disease space, things like herpes and an antibacterial project. I've worked on so many projects I can't even remember them anymore unless I look at a printout of what I worked on, because as I mentioned, a lot of them wind up failing and you just forget about them. But certainly has been an exciting adventure. It's always something new to learn, and every time you change from, say, cardio to neuro or neuro to infectious disease, there's a whole new field to sort of learn on the job, and that can be very fun and ultimately rewarding, because as you work from one spot to the next, you're increasing your overall knowledge base exponentially, to the point where you get to be an old man like me, and you know hopefully a whole lot about a lot of different things. But still no success, so who knows.
Okay, well, I guess, yeah, we'll wrap it up now. So thanks so much. I don't know if you want to close out the meeting or.
Thank you.
So for any of the club members here, I was thinking that we could hold a meeting next week to discuss a possible partnered food drive with multiple clubs of Methacton. So if any of you are busy right now, the meeting is basically over. So thank you for everyone for coming today, and we look forward to seeing you all soon. And thank you again, Mr. Skudlarek, for your presentation.
Thanks for having me. Thank you very much.
Okay, Mr. Adolph, do you think I can end it now?
Yeah, you're good. I'm sorry. Yep, you're good. Good job, guys. Well done. Regan, your dad's awesome.
Thanks.